Rappin' with ReefBum is sponsored by Bulk Reef Supply and Ecotech Marine.
Hey everybody, and welcome back to another episode of Rappin' with ReefBum. I'm your host, Keith Berkelhamer. So today I have the pleasure of welcoming Dr. Ben Titus to the show. Hey Ben.
How you doing? Thanks for having me.
So for those of you that don't know, Ben is an assistant professor in the Department of Biological Sciences at the University of Alabama. Ben received the MASNA Graduate Student Fellowship Award at MACNA 2017 and was a speaker at the 2019 MACNA. But before we start talking with Ben, I want to take care of a little bit of business and thank our sponsors for the show, Bulk Reef Supply and Ecotech Marine. I really appreciate the support from both of those companies, and I also really appreciate all you viewers tuning in and supporting the show as well. So please, as always, spread the word and hit that like button so more people can find the live stream. And as per usual, I encourage everybody to ask questions in the chat. One last piece of housekeeping. All episodes of Rappin' with ReefBum are now available as podcasts on Spotify, Apple Podcasts, and Google Podcasts. So if you want to catch replays of the shows in the podcast format, check it out on those platforms. So Ben, man, thanks again for taking the time to join us direct from Alabama.
Absolutely, yeah. Thanks for having me.
So you've got a very, very interesting background. You know, you're a biologist, you're a scientist. Can you explain to us in layman's terms what your focus is on in terms of what you're studying?
Sure. Yeah. So, I think I kind of look at my research program in sort of two different ways. I think, broadly, I'm sort of a classically trained evolutionary biologist. And so I'm really interested in understanding symbiosis and understanding how biodiversity evolves within symbiosis. So understanding how species evolve that are symbiotic or mutualistic. And I'm really interested on tropical marine symbiotic relationships in particular. And so I focus my research efforts on tropical sea anemones and their awesome crustacean and fish symbionts. And so that is sort of the big, broad, sort of basic research program that I run at the University of Alabama. And then more specifically from an applied side, these are animals that are major targets in the aquarium trade and hugely popular in the aquarium trade. And so from an applied perspective, I'm really interested in, how many species are in the aquarium trade, in particular in sort of the organismal groups that I'm interested in. So these sea anemones and crustacean symbionts. There's a lot of undescribed species that are sort of floating around, and I'm sort of interested in sort of disentangling how many there are and sort of what their stories are.
And we'll certainly dig into some of the specifics in terms of what these species are that we might not even know about in our reef tanks. But how did you get all interested in this sort of thing? How did you find yourself to get into this field? Was it a love you had as a kid, just for being around ocean life and whatnot?
Yeah, totally. I grew up in the Midwest, so I grew up in Bowling Green, Ohio. So our family would take vacations to the beach, really the Atlantic coast. But other than that, I really was not exposed to sort of marine science at all. But, I think like a lot of kids just sort of had a fascination with the ocean, loved Shark Week, loved going to the library, checking out books on sharks. My two favorite books growing up were like sharks and whales and all that kind of stuff. And so, yeah, I think, I get my haircut now a lot of times and, I tell people, hey, I'm a marine biologist, and you get I wanted to be a marine biologist too when I was little. And it's just sort of like one of those things. Yeah, it's like I never grew up essentially. I tried to. So I was a business major when I started college. I went to a small liberal arts university called Otterbein College, which is in a suburb of Columbus, Ohio. And I started as a business major and took an econ class, and I hated it. And in the meantime, I found out that there was a professor at our university in the biology department, actually a paleontologist. And he studied fossil coral reefs, and he taught a class called coral reef ecology. And that went to Belize every other year.
There you go.
And so I went to his office at the end of my freshman year, and I told him, I want to become a marine biologist. And, I'll never forget the first thing. He looked at me and he's like, look, there's no such thing as a marine biologist. He said, you are a biologist just like anybody else. You just happen to do research in the ocean. And so you should just get a general biology degree, and then just make sure that your research focus is on stuff you're interested in the ocean. And so I kind of did that. So I switched my major. And from there I was able to take his coral reef class, went to Belize, also did an undergrad research project with him that was on a fossil coral reef in the Dominican Republic, which was super cool. Still one of the more interesting research experiences. I love talking about that. And yeah, from there, I was sort of able to, use that research experience at Otterbein to sort of get into grad school, essentially. And then it's just kind of snowballed. And I've been sort of a transient academician from master's to PhD to multiple postdocs, and now finally kind of landing a permanent spot. But
how was the whole process of becoming earning that PhD? Was that a long haul?
It was. I did a master's first, which I always tell people that was the best thing I had decided to do. I honestly was not entirely sure coming out of undergrad that the PhD route was the one I was gonna take. And that sort of gave me sort of like a low pressure way to get into academia and to understand what research is all about and, make mistakes. You're 22 years old coming out of undergrad, and, you really know nothing. And so it gives you sort of a low pressure, low stakes way to kind of make a couple mistakes. And then at the end of that, I kind of had a clearer picture of what I wanted to do. So my PhD was great. I thought that was awesome. It was a phenomenal experience. And I did that at Ohio State University, and, about 5 years and some change, which is, pretty good for a PhD in the States.
Yeah.
But yeah, I've had a really good experience so far.
I kind of went the opposite route. I thought about biology in college, and then I took a biology class. I was like, that's going to be a little tough. Maybe I should kind of stick with the business side of things. So I ended up going to graduate school for communications and business. Spent like 26
Nice.
years in the media business, wearing a suit and whatnot. And
Yep.
about I don't know, 7 years ago or what have you, I had the opportunity to kind of make a life change. My wife and I became empty nesters, so I was like, you know what? Let's just kind of like drop the whole corporate grind thing. And we
Amazing.
moved up to Vermont, and now I'm doing what I really, really love.
Amazing. Amazing. Hey, everybody's got to find their path. I cannot imagine having to wear a shirt and tie. Like, I roll into work with a hat and
isn't it
t-shirt
awesome?
on. It's the best.
So we've got
Yeah.
Brymac1. The question is, I am a biology major. What kind of jobs are there out there for marine biologists?
It's a good question. There's really two kind of tracks. In my sort of estimation, you could sort of have the applied side, which, you could get a position at a state agency. So it's sort of maybe like a Florida Fish and Wildlife or, a state funded fish and wildlife position. There are federal positions that are similar in that vein with, NOAA. And then there's sort of like the academic research track. Which is more of like, getting a job as a researcher or faculty member at a university. All of those jobs, for the most part, are going to require an advanced degree. So, unfortunately, we've sort of reached this point where you need to go get a master's degree minimally, and a PhD would be nice in a lot of respects. I know a number of people at Florida Fish and Wildlife who were able to get in on the ground floor a number of years back with either a master's degree or without a master's degree, and they are now going to get an advanced degree while working simultaneously.
That's tough.
Because they feel like they need that to kind of keep their career moving a little bit. So, that's not the greatest,
Probably not what he wanted to hear
necessarily.
or she wanted to hear, but
it's, what, honestly, grad school is a phenomenal experience. It can be if you end up in the right spot. And yeah. So, and then there's the aquarium track too. And, honestly, and I think this is something that I find super fascinating. And one of the reasons I've loved staying in the aquarium trade world is because you get people like yourself and lots of other people that I've met over the last 4 or 5 years that have this sort of entrepreneurial, scientific, technical aspect that they really bring to the table. So the aquarium side of things is much more of like the Wild West from a professional standpoint. There's not one track, you don't even need a biology degree.
Right.
So, So, if you like that kind of thing, then there's sort of a non-traditional track. Like there's no degree in aquarium trade. So
you mentioned the aquarium trade. You actually worked at a coral farm, right, before you went to get the PhD?
Yeah, I did actually. So, Todd Melman runs this place out in New Albany, Ohio, named Reef Systems Coral Farm. He was looking for somebody right when I was finishing my master's degree at Auburn. And, I had defended my thesis. My wife was finishing her master's as well. She could have finished remotely. So we actually just moved back to Columbus. And I worked for Todd. And that was a phenomenal experience for me. It was a crash course in really understanding what it takes to maintain a tank, and quite frankly, just understanding the diversity and the sort of the scope of the trade itself. We're mostly a coral farm, so he actually had like this agricultural certification in Ohio. But then we'd bring stuff in from overseas as well from time to time, and then he would try and farm those. And, I did the service. So I drove the van around. I did all the service for Todd for, each week. I was doing probably 2 or 3 days out on the road, driving around to his different accounts. He was actually a co-PI on a National Science Foundation grant with Dr. Andrea Gertoli at Ohio State University. She's a coral biogeochemist. So we also had a big NSF funded project at the coral farm, which was really exciting to be around. And I also think it kind of gave me that perspective, like this is how these things can be married together in a really successful way. And it was really an interesting
Education there.
year.
Yeah.
Really. Yeah,
Yeah.
totally.
All right, man. So Ben, you gave a very interesting talk back. I mentioned you spoke at MACNA in 2019, and it was about these cryptic, undescribed species in our saltwater tanks. Can you just explain to the folks what that really means in terms of what your definition of cryptic species are?
Totally. You know, the best example, and I think I gave this in the talk, is mentioning the giraffes. So giraffes are described as a single species. They all look the same on the outside, essentially. But when you start to sequence the DNA of all the different giraffe populations in Africa, it turns out that there's actually 5 different species of giraffes that have all that genetically are very distinct. And they've been basically split from each other for 2 to 3 million years. So morphologically, physically, the physical characteristics, they look almost identical, but genetically they're very different. And so that's essentially what a cryptic species really is. We sort of call them cryptic species complexes because that's sort of implying there's more than one species. And it's the same thing for a lot of these invertebrates. So lots of these invertebrates, corals, shrimp, you name it, these are fairly simple animals from a morphological standpoint and very challenging to work with. There's not necessarily a physical characteristic that we can always use to differentiate between different species. But when you start to sequence them, then you start to realize there's a vast amount, there's way more species level diversity here than what's really just been described using just the morphology alone. And so that can take something like, one of the animals I worked on is the sexy shrimp. It's one of my favorite, Thor ambonensis.
Gotta love that
that's
Latin
one
name.
species. Absolutely. And so you have one described species that's global, and when we sequenced it all, it turned out that it's at least 5. And so that's what we're kind of dealing with. And then since the trade is bringing in animals from all over the world, you really are into a situation where, you really probably do have dozens and dozens of species that are undescribed in everybody's home tanks.
So what are the implications of that for the sexy shrimp? Would you expect to see different types of behaviors for those different species of sexy shrimp?
From a behavioral perspective, they all seem to occupy the same sort of ecological niche space. So they live on anemones and they live on corals and things like that. They all wiggle their butt in the air. So
I have to pick up one of those.
So from that standpoint, yeah, exactly. So from that standpoint, not really. But the implications would be if you want to breed them, for instance. Maybe you're having a really hard time and you want to start an aquaculture, and lots of people like to breed their own animals at home, and you're having a tough time trying to get these two things bred. Well, you might have a species from Florida, which just got redescribed. It's the first one that's been redescribed. It's Thor DiCaprio from Leonardo DiCaprio is like the inspiration behind that name. And then you might have a species from Indonesia. And, it's possible if you want to do an aquaculture program, you're just dealing with 2 different things that might not reproduce. And then, the behavioral side of things, and obviously the conservation side of things. I mentioned some of those jobs at Fish and Wildlife. They have people there that are there specifically to, manage the trade and make sure that harvest quotas are being met. But if there's all sorts of undescribed species, it's hard to really manage it effectively.
So, all right, you're saying that behavior might not really be noticeable between different species of the same invertebrate, let's say. So, for instance, the reason why I'm asking this question is peppermint shrimp, right? A lot of folks will use peppermint shrimp to fight aptasia in their reef tanks. But I've heard that certain types of peppermint shrimp might not necessarily be prone to eating aptasia versus other types. Is that because there might be, cryptic species within the peppermint shrimp family, or is that potentially not related to that?
Yeah. I don't want to give the impression that there are not behavioral differences from a cryptic species standpoint. Specifically with Thor, I didn't see anything ecologically, on all the diving that I did and all the collecting I've done that indicates that there is. I mean, there might be, if you look at it a little bit closely or maybe differently than I have. But to your point with, the peppermint shrimp, totally possible. I'm not sure actually if anybody's looked at that or not.
Hmm.
Whether there is cryptic. I know people have looked at like Stenopus, the banded coral shrimp, and some of these other cleaners, but I don't know what the research has been on, Lismata and some of the other peppermint shrimps yet. But it could be, yeah. I mean, it could be, preferences can definitely change, especially with speciation events, different environments, habitats, and that kind of stuff.
Do you ever get any information that you act on from the hobby about certain invertebrates that we keep in our reef tanks? I mean, if there are certain observations that are made by hobbyists about such animals, is that something that might be fodder for you as a scientist, a biologist, to kind of take that information and maybe study it, or is there a whole different process that you go through in terms of kind of like what is on your plate to dig into?
It's both, honestly, and it can be both. I find there to be a slightly, you know, a lack of communication, I guess, or maybe fewer avenues of communication between like pure academicians and the hobby that I think would be really beneficial if they were more open. Um, you know, a classic, honestly, a great example of this is with Thor. The paper, one of the papers that we had to cite forever was this paper from the 1970s where you have Thor was, the larval dispersal phase of Thor was like 50 days in an aquarium. And no, none of these researchers could get it to actually settle. Whether you put an anemone or a coral in the tank, it wouldn't get these larvae to settle. So they just assumed that, it has a massive range, it's globally distributed, it's got a massive larval duration in the water column. And then you go on some of the reef tank forums and they know exactly how many days it is until Thor goes through metamorphosis and settles. And then a couple of years back, another, paper was finally written that pinned it down, and the trade had that sort of figured out 15 years earlier.
Yeah.
And so, I think definitely I want to hear these kinds of things. I think it's really interesting. It's hard to know, what, thread to kind of follow at times. Individual projects. I think the way science is done a lot of times, it's not so much individual projects like, hey, I saw this really cool thing, this really interesting behavior that this happened with this one shrimp. Can you tell me if this is interesting or not? Our funding usually comes from sort of conceptualizing really broad questions and then
Yeah.
sort of having a multi-year investigation. A lot of times we don't have a lot of sort of discretionary funds to just sort of like,
Yeah.
go down the rabbit hole, even though that would be nice.
There's a lot of rabbit holes in those forums for sure.
There's a lot of rabbit holes. But with my question with the sort of species level, how many species there are, that is a question that, right now I have funding from the National Science Foundation to do the research into the clownfish hosting sea anemones and figure out how many species there actually are. Part of that is to get samples from the trade and to understand that. And so, you can send out my email address, and if anyone wants to send me samples, I'm willing to take them. So that kind of stuff I'm definitely open to and interested in. I do have some funding for that.
Yeah, and I definitely want to talk more about sea anemones with you because you're pretty much like a sea anemone expert in terms of the symbiotic relationship between those invertebrates and clownfish. But one thing that is interesting to me, I'm a big SPS nut, so I love the sticks. I've been keeping like SPS dominant reef tanks for many years, but there's I believe there's some research out there about acro-eating flatworms, some actual academic research about the life cycle of acro-eating flatworms. But that would be something interesting to to see even more research on in terms of the life cycle and how to nuke that type of pest in a reef tank, because it is tough, man. It is really tough.
Yep. Yeah, absolutely. And things like that are sort of these sort of like very applied questions. If there was a company, that or an agent, something, any private foundation that wanted to fund that kind of work, I guarantee you could find a flatworm specialist. Let's rear these things. Let's kind of figure it out. But again, the lines have to be open and people have to know who each other are. And there has to be some, legitimate mechanisms behind that. But yeah, there's stuff like that.
I mean, some of the the research that's developed in the in the trade over the years has been pretty amazing. In terms of like red bugs. You use bear to or I'm sorry, interceptor to
Yep.
dog heartworm medication. Somebody figured that out. That will
Yep.
kill the red bugs, but nothing else in the tank except for, I guess, crustaceans. And now
That's right.
somebody figured out to use bear insecticide to kill acro-eating flatworms. So somebody's doing the research on the hobby side, and
That's
there
right.
is academic research going on. But yeah, it would be great to
Yeah.
kind of like see some sort of tighter lines in terms of the back and forth
Hmm.
communication.
Yeah, definitely. And someone like Andy Ryan at Roger Williams, like obviously does a phenomenal job of linking those two. And there just need to be more people, I think, on both ends of it.
So Ray S made a comment about his sexy shrimp. Says it likes hanging out on the mini max, seems to all disappear after a few months, and the question is, what is their lifespan? I think you have answered that question, right? 5 or 6 months, or was that 5 or 6 weeks in terms of the
Thor?
yeah.
That's a good question. Honestly, I don't really know. A lot of these shrimp I think they have pretty short lifespans. Um, there's one shrimp that we work on. It's a cleaner shrimp from the Caribbean, Peterson's cleaner shrimp. It's one of those really pretty purple cleaners. That thing reaches sexual maturity at 6 months old. And so if that's quick. So, the anemones that it lives on in the Caribbean only live for about a year and a half, 2 years tops. So you're talking about a shrimp that can recruit to an anemone, gain maturity in 6 months, and start reproducing. And yeah, so I would guess less than 2 years would be my
So this
guess.
is an interesting question from Kia Aloha Reef, I think is how you pronounce that. To what extent are you witnessing species adapting to climate change?
It's a very good, and I am not a climate change researcher, so that is a difficult one for me to answer from a position of expertise. But I think we're seeing ranges expand in a lot of
Hmm.
species. I think that is something that's been seen quite a bit. There's some cool anemone observations that we're starting to see some really big shifts, or at least just some increases in population size and maybe sort of the edges of some of those ranges. Um, but yeah, that's a difficult question to answer, and it's difficult to disentangle a physiological adaptation that an individual would have. If you get hot, you sort of ramp up these sort of heat stress proteins or things like that, versus like a species level adaptation, which is like selection is acting on genes that are beneficial because it's getting hotter. And so, there are other people that are doing that kind of work. But, we know about evolution. We kind of understand about how evolution works. So there are definitely things adapting to climate change. Whether they can adapt fast enough to stick around is another question.
Yeah, have you found the some of the coral reefs that you've been working on to be impacted pretty badly by climate change? I mean, are there certain areas of the world that are just becoming less and less accessible to researchers because of that, or is it actually providing an opportunity to learn more about climate change?
Yeah, it's going to be both. I mean, I was out in Moorea in French Polynesia a couple years back in 2019, and I was out there collaborating with a research group that works on anemone fish specifically because anemones in the lagoon, some of them bleached during the last heat wave, some of them didn't. So they did some moving around, and we did some work on them, try and figure out the genomic basis of it. And we've not really generated those data yet. But that whole reef after I was there bleached like 2 weeks later,
Ooh.
and yeah, so Moorea and French Polynesia have been hit by a lot of really bad bleaching. The reefs there seem to be a little more species poor. They're not as diverse, and so if you have a sort of species poor reef and everything responds in a similar way, you can wipe it out really quickly. Um, yeah, so it's an opportunity as a scientist to understand it, but it's a little depressing.
Yeah, no, that is a little depressing. So, all right, invertebrates. That's really the thing that you're been studying the most in terms of the cryptic species.
Yep.
What about fish? You know,
Mhm.
I know you're not an expert in terms of cryptic species of fish, but can you talk to that a little bit? Are there cryptic species of fish out there that are in the aquarium trade that we might not know about in our tanks?
Yeah, almost certainly. And I would not be surprised if some of them are clownfish. And, some areas of the world are really hard to get to. Um, Indonesia, for example, there's lots of stuff that, has historically come out of Bali. I don't really know if Bali has opened back up. I know there was a big shutdown there recently. But if, as a scientist, if you want to go study anything from Indonesia, it is almost impossible to get samples out.
Hmm.
And even if you have a local collaborator, it's really hard to get samples out. And so a lot of times when we do some biogeography where we're sampling across a huge area, you look at these papers and Indonesia is just empty from a sample perspective because it's almost always off limits
Hmm.
to research. And there's a long history of colonialism and all sorts of stuff that has gone on that sort of shape the policies of these countries, but it
Yep.
doesn't make it any easier. But I mean, there's like all sorts of really interesting morphs that are found and pop up all the time especially from the fish side. The Clark eye, the Clark's clown anemone fish or clownfish. Lots of debate about how many species there are. It's probably at least two. Even within, Ocellaris, you have like the black Darwin clown.
Yep.
That looks like it's genetically distinct. I think there have been some interesting things with like the maroon lightning clown that's been shown some interesting genetic patterns as well. And even the true Percula clownfish. There's all sorts of interesting little, morphs of clownfish. And, no one really has a great answer for it yet. And some of these places are just hard to get samples.
I wonder what the scientific community's reaction is to all the breeding that's going on in the aquarium trade in terms of clownfish. I mean, there are just so many morphs of clownfish out there, and I just wonder what that's doing to the clownfish. The different species of clownfish out there in terms of all that. I mean, there's got to be a lot of things going on in terms of that kind of breeding.
Yeah, you would think there would be some really interesting like genome rearrangements and combination of genes that you wouldn't get in the wild necessarily. But I think most of the people are probably pretty happy that that's going on. So then you keep the wild fish in the wild, and then you can breed them to your heart's content in a tank. And I think that I think that's a positive, by and large.
All right, so you've been talked a lot about sea anemones, and that's an area of your expertise. Can you just discuss a little bit in terms of the kind of work that you've done on that side?
Yeah, absolutely. So, tropical sea anemones are sort of interesting. We think about the clownfish as this really well-known symbiosis. Clownfish are the poster child for the aquarium trade. It's probably one of the most well-known animals on the planet, really, with Finding Nemo and Dory and all the movies.
Yep.
And so, I think people are pretty surprised in a lot of ways when you tell them, we just really don't know anything about the sea anemones. And not only that, but tropical sea anemones really haven't had a whole lot of attention since really the advent of modern DNA sequencing. So, that is a long time ago. So we are playing serious catch up. And there are a number of reasons for that, why they've sort of been overlooked in a lot of ways. Some of it just has to do with they're a challenging animal to work with. So early on, a lot of the DNA sequencing approaches were using mitochondrial DNA as these sort of DNA barcodes, which have been useful for figuring out, hey, this might be a new species, or this is the same, this is different, those kind of questions.
Yep.
But for whatever reason, sea anemones and corals fall into this category. Their mitochondrial DNA evolved so slowly that we can't use those same kind of markers that you can use for a shrimp or a mammal for the anemones. And so that's put us behind the 8 ball. And, for basically 30, 40 years, we just have been very limited in our ability to figure out how many species there are. And, when we're talking about the clownfish symbiosis broadly, that's 50% of the interaction we just don't have any clue about, relatively speaking. So we think we might have this idea about how the whole symbiosis has evolved and, these behaviors and host specialization and what is really going on in the system. The anemone is just as important as the fish to make the whole system work. And we don't know anything about it. So yeah, for the last 3 years or so, I've really been working on that side of things. And so we've, really started from the bottom. We've published a paper showing that the symbiosis has evolved 3 times independently with sea anemones. So clownfish, right? They're this group of 28 or so described species that all evolved from a common ancestor, whereas the anemones actually are, 3 major independent lineages within the broader sea anemone order. And so, either the fish have evolved to live with lots of different anemones and vice versa. So there are these independent evolutionary events that have happened in the sea anemones. And so now we're really moving on from there, really trying to figure out how many species belong to each of these 3 groups. And so one of those big groups is the bubble tip anemones. That's described as a single species. A lot of the new data we're generating is looking like it's probably going to be 4, maybe 5 different species of bubble tip anemones alone. And so if I had to guess, by the time it's all said and done, there's 10 described species of anemones that host clownfish right now, and it's probably going to be 20 or more.
Wow.
Would be my guess. Yeah.
So what are the implications for us as hobbyists? With that sort of information in hand from the scientific community, would that allow us to make better decisions in terms of pairing clownfish with anemones?
I think that's a good question. It's hard to say because you have like Ocellaris will live with like a Caribbean anemone from time to time. And so you put a fish in a tank and they behave very differently than they do in the wild.
Right.
So from a hobbyist perspective, it's unclear how that would really change these interactions. Amphiprion ocellaris, the Clark, or the Ocellaris clown or the Percula clown, those are species that never associate with bubble tip anemones in the wild. And they do all the time in a tank. So how that affects the interaction might not affect it all that much from a hobbyist perspective. But if I know the hobbyist, I know that everybody likes the coolest, rarest, most unique thing.
Yeah.
And so, that's sort of a thing. And if there's a really rare local endemic someplace, bubble tip anemone, the Colorado, I was at the last MACNA that we had in person, it was the Colorado sunburst.
That's crazy price.
And, all these things, that's crazy prices. And those are just different colors. Imagine if that's like not only a cool color, but that's like a new rare species. And, that'll send that thing through the roof.
Yeah.
And so, yeah. So it affects the psyche. It doesn't necessarily affect the interaction with the fish.
It's interesting because, I've had many clownfish and I've never kept anemones. Well, I take that back. I once had a bubble tip anemone and it lasted one day because it got sucked into the power head. So I lost that bubble tip anemone, and I've always been hesitant to have anemones because I like SPS and they tend to wander around the tank. But there's a lot of folks out there that have fine luck keeping anemones with the SPS dominant tank. But I've found them to host like a leather coral. I've found them to host in a clam. Torch coral. Pretty much right now I've got 2 that are living in a Goniopora, and nice. But it was weird because they both were not mated a pair of clowns. I got them separately, and for like the first couple of years they slept in different corners of the tank, and then all of a sudden one day I noticed that there was a clutch of eggs. I was like, how'd that happen?
Oh, interesting.
Yeah,
that's really interesting. It's one of the honestly, the single biggest mystery with the clownfish stuff is what is the mechanism that allows these fish to live in the anemone? We still don't actually know that. There's some pretty good hypotheses. about maybe it's like chemical camouflage, right? There's some sort of like convergence between the mucus on the fish and the mucus in the anemone, and the anemone just sort of is recognizing the fish as self. But, you know, from a fish standpoint, like there could be some sort of like, you know, tactile stimulation that they are gaining from the anemone that's sort of that they like. So if they find something that can provide like a similar tactile sensation, then maybe you can get like what you're saying, like a, you know, a mated pair and a goniopora.
So, folks, don't be afraid to chime in here. We have a sea anemone expert with us and an invertebrate expert. So feel free to fire away with some questions in the chat. I've definitely got some more questions there for you, Ben, because I find this stuff fascinating in terms of trying to understand the behaviors of these animals that we keep in our tanks. And I guess there are different behaviors that you will witness in captivity versus in the wild. And is that something that you have to factor into the research that you do? Because I'm assuming that you don't always get the chance to study these things in the wild.
Yeah, you know, I think that's a huge one. If you ever want to do sort of a manipulative lab experiment where you can actually control for a lot of different variables that you want an answer, like, hey, what does the microbiome, so the microbial community, do with a fish and without a fish? Well, if you put a clownfish in a tank, you know, sometimes they just don't even go in the anemone at all. So it's hard to really control for those factors, and you have to really take that into account. And it can make doing laboratory work really problematic. If you want to understand like host choice, for instance, that's a really important perspective from the clownfish evolution. It's like, what fish or what anemone does it prefer? Well, if you take it out of the wild and put it in a tank and it lives with a goniopora just as easily as it does with a bubble tip or anything else, then your conclusion is, well, host choice isn't important. But if you go on a reef, there are very clear non-random patterns that you see in the wild that are important. And so it's hard to really tease these factors apart. And part of the reason why right now I'm really good at going to the field and collecting and killing things. And I'm not very good at keeping them alive yet right now.
Oh.
It's a lot of work for maybe not as much payoff.
Yeah. So we have a question. I'm not sure you're going to be able to answer this question or not, but we'll run it up the flagpole.
Sure.
Ghouls is asking, I'd like to add another clownfish to my one lonely clown I have now, and any special tips on how to add it? Should the patterns be similar size, etc.?
It's a good question. There's probably someone more experienced than I am, but I've always been under the impression that if you add similar sized clownfish, then things can get a little dicey.
Yeah.
So I might add a smaller one. Does that sound about more in line with your experience?
Yeah, I think so. Yeah. You know, and I just about a year ago I started a new peninsula tank. It's a 225-gallon peninsula tank, and it's 6 foot long by 3 foot wide, 20 inches tall. And I love clownfish, and I've always had just one pair of clownfish in my reef tanks. And the biggest tank I ever had was a 225-gallon tank in Connecticut that was broken down many years ago. So, but I was determined to get 2 different pairs of clownfish into that new tank. And knock on wood, so far so good. You know, I had 2 different pairs that I had in 2 different frag tanks that just were hanging out solo, you know, in terms of being in those 2 different tanks, those 2 different pairs of clownfish, and I don't know, for 6 months to 8 months or whatnot. And then at the same time, I put both pairs of the clownfish in the new tank, and it's kind of a weird thing that's going on right now. But you know, at night they kind of like all sleep together in the same bunch in one corner of the tank, but then during the day when the lights are on, they're at opposite ends of the tank. So
very cool.
Yeah. But you know, I guess that's a whole other consideration in terms of the size of the territory with clownfish.
Yeah, absolutely. Are these all ocellaris?
So one pair is definitely ocellaris, and the other are the Mocha Storm. I'm not sure if those are ocellaris or not. Do you know that?
I think they are.
Yeah,
they're dark, like a darker pair.
Yeah.
Okay.
Yeah. Okay, we've got a whole bunch of sea anemone and clownfish questions popping in
All
here,
right, here we
Ben. So
go.
hopefully I didn't put you on the spot too much
I'll
here.
do my best.
Braveheart Reaper 525. Why do bubble tips lose the bubbles in aquariums?
Another great question. That is one of those things I've always wanted to know the answer to that. And from what I have read and talked to people about, people think that is some combination of the water flow in your tank and the light in your tank. And so I don't really have a specific answer about what factor controls what, but anemones are very delicate. So um, if you have a really high flow tank, I would imagine, scrunching up your tentacles, creating more bulbous, like less drag. If you have lower flow, you can envision a scenario where the tentacles elongate some and might increase a sort of catch radius from a prey capture perspective. And I've also heard the light's important. So like maybe the bubbles do something by refracting light in a way that's, increases the efficiency of the symbiodinium, the symbiotic algae that live with them. It's a good question. I'd love to know the answer to that too. I also think the fish is important. I think, one of the papers that we did, I was a master's student in the Red Sea. We found these bubble tip anemones. The pattern was that they only had long, sort of finger-like tentacles when they had two adult breeding pairs in them. So the ones that were, stubbier, more bulbous anemones had, either a juvenile in it or just a single adult or a subadult. And so I think that's a factor too. I think the fish presence could potentially be a factor.
What about mixing different types of clownfish? Deep Reef has got a question about that. Can a Percula and Ocellaris pair up? I mean, in your research, have you got into that as a variable in terms of the symbiosis between a sea anemone and a clownfish pair? Or is it pretty much, you're looking at one species of clownfish and one species of anemone to study? Or have you ever tried to kind of flip it up a little bit in terms of the clownfish?
Yeah, I think I know of some data that will be coming eventually from Switzerland. So I was in Switzerland previously in Nicholas Salmon's lab group. They're using the full genomes to look at, how hybridization has shaped speciation events in clownfish. And it's important at some level for some species. And so there's definitely been some, hybridization in the past. But yeah, that's a good question. I don't know. I mean, I think, hybridization events, if they like actually occur, are going to be basically, by definition, going to probably be pretty rare. So I think they're much more likely to probably fight each other than mate with each other or coexist. And there are some really rare patterns that you can see in the coral triangle where you do get multiple species living on the same anemone. And that's in a really sort of densely packed region with a lot of different species of clownfish, where there are more species of clownfish than species of anemones. And so you do see some interest occasionally where you will get that. But let us know how it goes.
So Ben, you mentioned Switzerland, and before we went on the live stream, you were telling me about what was going on in Switzerland. Can you share that on the stream in terms of what the work you were doing in Switzerland? And to me, it's like, wow, you're going to Switzerland, and you're studying clownfish.
Yeah. So before the year before, so my family and I were in Switzerland last year. Moved there in August of 2020 and then moved back at the end of June of this year. And, I had previously written a 2-year postdoc proposal to the European Union for a Marie Curie fellowship. And Switzerland's obviously not in the EU, but they're part of the sort of broader EU economic zone. So I was able to take that fellowship and take it to Switzerland. And there's a professor there, Nicholas Salomon, who he's the chair of the Department of Computational Biology at the University of Lausanne. So right on Lake Geneva. Is beautiful. And he is an evolutionary biologist who has been really leading the charge in a lot of ways with doing the full genome sequencing and evolution of the clownfish. And so their lab has done a lot of really nice stuff, and they've sequenced a number of different clownfish genomes. And as a sea anemone biologist, I really hadn't had an opportunity to do the full genome work yet, and so I wrote a proposal to do the full genomes for the clownfish anemones. And so that was ultimately funded, and so we got to go live in Switzerland. So that is really that's work that I'm now doing at Alabama. So we're going to be sequencing the full genomes of the anemones, and then using those genomes to ask some questions about mutualism and how the mutualism with clownfish has affected the genomic architecture and evolution of the anemone genomes. So getting at a number of different types of questions that are, again, this is a very basic research kind of question rather than sort of an applied stuff. But, things like the fish are providing really important services to the anemone too, right? So they're fighting off predators. They're constantly the anemone as they move through the anemone. They're facilitating gas transfer, so they're sort of oxygenating their host. Those are all really important things for the sea anemone that could ultimately allow them to evolve different characteristics. Maybe they evolved a really large body size because they had this symbiosis with the fish that allowed them to do that. Things like that.
So NSB Reese has got a question, and it's an interesting question, but the direct question is, how does calcium play a part with anemones? Does that impact their behavior at all, their health at all, in terms of tank parameters?
I don't know. If I had to guess, I would imagine it's a very limited effect. Anemones are going to be a lot more flexible in terms of their environmental parameters than an SPS or an LPS coral will, and you see that too in the wild. They live farther north or farther south than like a reef. So as a reef peters out, you'll get anemones.
What I don't know if you'd be able to answer this question, but what kind of tips would you provide to someone that wants to keep an anemone in their reef tank in terms of the optimal conditions for a sea anemone? Obviously, I think to have clownfish hosting would seems to help. But any things to general tips for sea anemones?
That's a good question. Let the thing settle and find a nice home. I think the water velocity in a tank is probably going to be pretty for the anemone.
Yeah.
I know a lot of people hate having them because they move all over the place. If you're trying to grow a bunch of SPS corals and you're blasting, like an ecotech pump and it's really churning the water around, I doubt an anemone is going to be super thrilled to hang out in a tank like that. I mean, but for the most part, they live right on the reef with everything else. So I think you're going to end up with a lot of the same kind of environmental parameters that they require. They can, if you have a nutrient bomb of a tank, they're probably going to be happier than a coral will be. But I think the water motion is probably a really important one. They'll move around until they find that nice hidden spot.
And would a mature tank be better than versus a young tank to try to keep an anemone?
I don't know. That's a good question. I don't know. Maybe it'd be better to do a young tank and then you don't, it doesn't kill all your corals. Let it settle and get in there first, and then you can add corals around it after it's kind of found a nice spot. That would be my guess.
All right, a couple more questions here, Ben. Sure. How are carpet anemones collected? I don't know if you have any info on that. Do they propagate easily? I'm up in Canada, and their prices are extremely high, $500 to $600. Just wondering if they are hard to collect.
Yeah, they're hard to collect. They're huge. So the ones that we really are only seeing in the trade are, Stichodactyla haddoni, which is the smallest typically of the carpet anemones. Stichodactyla mertensi it's a meter across on the reef. It's just like drapes over the reef. So you're not going to collect big animals. So, from a collecting standpoint, you need to find, fairly young, recently settled individuals to actually collect reasonably in the trade. And there's a sexual species only. So they are not propagating asexually on the reef, so they're just going to be at lower abundance, and I think that's probably why you're seeing the price where it's at.
So another question from Matt A. Will decades of propagation in the aquarium trade / home aquariums alter the genome of corals making them a different species from the original wild colony. I guess in terms of corals, that might not be your area of expertise, but any thoughts on that question?
Yeah, I mean the propagation, the mutations that you're going to get by propagating things asexually are going to be, we call those somatic mutations. So those are not mutations that are being passed down generationally through sexual reproduction, and so those are not rearranging the genome. And so somatic mutations are important, especially in terms of like, an individual lifespan. So maybe it, induces senescence, which is just this naturally occurring process of things dying. So maybe you get a bunch of bad mutations accruing in a colony or a genetic line, and then it just dies off. But if you think about dogs, for instance, and how those have been sort of artificially bred by humans, essentially into a new species, you could presumably do that if you can do it through sexual reproduction. So
yeah,
lots of examples of domestication.
There you go.
Yeah.
Another interesting question from Drama D. Curious if tank temp plays a role. Cold water anemones, temp tank comparisons would be a cool study. How low can a bubble tip go? Type of thing. I guess. Yeah. What's the impact of temperature on the sea anemones?
Yeah. It's a good question. So I have some collaborators in Japan, and they've done some phenomenal sampling for me through a project that we have ongoing. And they've sent me some really cool pictures of some of the sites they've collected from. And one of them is up in the northern part of the Japanese islands, up near Tokyo, which is a very temperate, almost cold water area. And there is just carpet of bubble tip anemones as far as you can see. There are no fish. There are no clownfish anywhere in those pictures. And there's like fronds of kelp that are like growing up in between some of the anemones. And it's really interesting. It's almost like, if you went diving off, I don't think it's quite as cold as like diving off the coast of California. But yeah, I think they can tolerate much lower temperatures. I cannot imagine them tolerating much lower than like 65 degrees. That would be my guess. That'd be pretty cold.
Yeah, that is pretty cool. That would be a cold water reef tank for sure.
Yeah.
Here's another interesting question from Ghouls. Do anemones carry any pests? I don't think I've ever seen a dip for them. Anything to worry about when adding straight to a tank? Thanks for all the great info.
Yeah. Well, yeah, happy to be here. Great question. Don't know. Don't know.
Yeah, I've never seen anybody dip an anemone.
Yeah.
Probably don't want to do that.
Probably not. I would stay away from that. If you're really worried, set up a little quarantine tank and see what happens.
Yeah. So the shrimp might not be helping in terms of any parasites that might be on the sea anemones or whatnot. Those are more about the symbiotic relationship between the cleaner shrimp and a fish versus an anemone in terms of the pests.
Yeah, absolutely.
Okay. All right, so Ben, you're a biologist, and last week I had another biologist on the show, Dr. Eli Meyer, who started up a new company.
Oh, nice.
And I don't know if you've heard of Aquabiomics, but they're a company that does bacteria testing. So hobbyists can send in, water samples, and they can test for the actual DNA in the tank to see what you have in there in terms of pests or other, critters, I guess. And then
very cool.
They could also study the, you mentioned microbiome, so they can kind of what kind of bacteria, you have in your tank, whether it's good and bad bacteria. Do you think we're going to see more science-based services like this become available to reef keepers in the future? Because I don't know of anybody else that's doing that sort of service. And you mentioned before that the lines of communication between the trade and the scientific community are not terribly tight, but yeah, just kind of interested in your thoughts in terms of will we get more science available to us in terms of the tank? I mean, ICP testing obviously is something that has been great in terms of being able to analyze the water, but anything else that you think might be coming down the pike?
Yeah, I think those are great services. I hope they become more popular. I've always been interested, understanding like what is the reef tank microbiome. I was able to do sort of a microbiome paper on the anemones that host clownfish in the Maldives. That kind of got me dip my toe into some of those methods.
Yep.
And yeah, it's critically important. And so I think, people are starting to talk about reef restoration in terms of like restoring them, like adding microbial, like healthy microbial communities to like wild reefs to help it maintain its health and all sorts of stuff. So I would imagine that there is a huge potential demand for that kind of thing in the hobby. If we, but we need the basic research first. We need to understand, what does, your typical good bacterial community look like in a reef tank. I think we think of these tanks a lot of times as like mini oceans, but they're just not. It's very different. There's even the debate between sand, having a sand bed and not having a sand bed. And you'll hear people like, well, there's sand in the ocean. It's like, well, there is, the volume of sand to water in the ocean is not even comparable. But in a tank, it's a huge deal.
Yeah.
So, those are important considerations to take. So I think that stuff is really cool.
So Drew Young has another anemone question for you there, Ben. What are the best propagation tips for anemones? Many hobbyists force a 50% water change to force a split after months of feeding. Any tips on propagation?
That's a good question. Some people just cut them in half. So that's, Colin Ford, the coral morphologic guy, he does a lot of cool stuff. He'll just cut Phymanthus in half. And that's not an asexually reproducing species. And he is able to do it sometimes. With the bubble tip anemones, you could probably cut those things in half. And because they do split. But I know people who have anemones just that are splitting left and right, and other people, who their anemones won't split at all. So I actually don't really know the answer to like what triggers it specifically.
Well, Drama D said that somebody had a power outage and interrupted the light schedule, and all of a sudden the anemone split, and then apparently were pissed off. So I guess sometimes you can go figure.
There's a long history of there's this theory behind reproduction that was back in the 60s and 70s. It's called the strawberry coral model, and the question essentially is, when do you asexually reproduce versus sexually reproduce? And one of the theories behind the asexual propagation is, when things are going really well, you split, because why would you sexually reproduce and lose half your genome to the next generation when you're going really well right here? And the flip side is, when I'm really stressed out, you should split. And so, I don't think there's ever been any real resolution there. So it might be stress.
So Deep Reef is wondering, what are some anemone diseases to be aware of?
Diseases?
Yeah.
That's a great question. Honestly, I don't really know. I don't know if there's really any research. Again, there's like a handful of us in the whole world that are really studying sea anemones, and
but you got a lot of times, you got a lot of people that are giving you fodder for studies right here.
I know. Well, the disease thing's really cool, and every now and then you'll see some anemone that looks a little gnarly. The tentacles look splotchy and bad, and it's a good question. No one's really working on it. And the other problem is, these anemones, they don't have skeletons. When they die, they just die, and then they dissolve and they disappear. And so there's not a lot of opportunity to really catch it while it's happening. And I think that's part of it too. You can see black band disease eat away, a coral colony for weeks maybe, but an anemone is going to die and dissolve and disappear in like a day.
So what drives the decisions to come up with a study, a scientific study? What are behind those decisions? Because obviously the aquarium trade is the aquarium trade, and I would assume that there's not going to be a lot of influence by the aquarium trade on the scientific community to study something, unless there is going to be some sort of advantage for both sides. But what kind of like drives the the different types of studies that you are involved with in terms of that kind of work?
Yeah, it's a great question. We try and link a lot of the research we do to like some really broad conceptual ideas. As much as it pains me to say this, because I consider myself like an organismal biologist. Like, I love sea anemones and I love the animals themselves, and I got into this because I loved these animals and I just wanted to know more about them. And the reality is a lot of people in the sciences, like another evolutionary biologist, doesn't care about sea anemones. Like I don't care about their birds or their snakes or whatever. You know what I mean? There's just not this like inherent pull to like need to know about, oh man, I can't wait to find out what's going on with the bluebirds or whatever. And so in order to sort of like garner broader interest is that you have to stay broad typically. And so, what are the questions if you're interested in like bird speciation? Well, maybe there's some really cool parallels in terms of the way birds speciate and the way anemones speciate or the way corals speciate or the way snakes speciate. So we try and stay kind of broad conceptually with some of these questions. And it might be like the role of allopatric versus sympatric speciation or the role of gene flow in the speciation process. There's lots of complicated ways things can speciate. And so those are ways that we can kind of connect the dots, reach broader scientific audience. And that is really important too from us from a funding perspective, because, the National Science Foundation, some of these agencies that fund basic research, they want it to be as broad and as applicable as possible. So we really try and work to make sure that our proposals are broad, they're interesting to the broader community, and that we're going to learn something that the broader community can then incorporate and build off of. So, we think about science as sort of this process of building on each other. And so it's sort of, that's a big motivating factor. So yeah, unless you work on humans, and then you can just say humans are interesting because we're all human. They don't have to do that as much.
Right. Yeah. Now listen, man, I think it's really cool stuff that you're one of the few people in the world that are studying this sort of thing. And that just kind of goes to show in terms of like how many different, just in terms of what the questions on the minds of the viewers here tonight, in terms of and some of the stuff that you guys haven't looked at. It's just amazing to me that there is so much to be done on that end, and so few people out there that have the ability and the funding and the capability to do that kind of research.
Yeah, absolutely. And a couple ones, obviously, that the trade, I'd love to hear from people in the trade that have, anemone lifespan. Would love to know more about that. Would love to hear even anecdotal evidence, how long your anemones, you've been able to keep them alive. Like that's a hugely important question that, we don't, have a good handle on. We know the fish can stay alive for 30 years. So presumably the anemones have got to be able to stay alive for a long time too. But, those are things that, we don't understand, but the trade would be hugely helpful, and, pinging us emails from time to time or asking us questions. And, I love to hear from people, cool observations like that. So if you've had a 20-year-old tank and, carpet anemone that's hanging around, that would be awesome.
Ben, is there a way for folks to find you out there on the web, or?
Yeah, totally. My
don't give out your don't give out your email address,
but yeah, my website is tituslab.ua.edu, and I am also on Twitter. It's at BenTitus4. I don't know where the 4 came from. It was just the auto-generated Twitter thing, however many years ago it was when I got the Twitter account. So yeah, feel free to reach out. I'm sure my email address is out there publicly somewhere.
Hopefully you're not getting.
Hopefully you're not getting. It probably has to be, because I'm a state employee.
All right. Well, hope you're not getting spammed too badly. But Ben, man, this was awesome. A very educational chat. Any last things you wanted to add before we sign off here?
I don't know. Hopefully, I'll see you all in person at a MACNA conference next year.
Yeah.
So I'll be there if there
that's
is
my.
I think it's Milwaukee.
Yeah, that's my plan, is to be in Milwaukee too. So hopefully I'll see you there.
Good.
Ben. Yeah.
Good. Absolutely.
All right. Well, listen, Ben, thank you so much again for being a part of this live stream.
Absolutely.
And I want to thank everybody for tuning in and asking some great questions. And as Ben said, please reach out to him on the web or Twitter if you have any intelligence that you could pass along, or any questions. But I also want to thank the sponsors, Bulk Reef Supply and Ecotech Marine, for supporting the show, and for everybody tuning in and watching. I also want to remind you folks that all episodes of Rappin With ReefBum are now available as podcasts on Spotify, Apple Podcasts, and Google Podcasts. My next live stream will be next Thursday, October 14th at 7 p.m. Eastern Standard Time, and it's going to be with Hobbyist Joe Muscat, also known as 2C on Reef to Reef, and he's got a pretty kick-ass tank, so it should be a great show. We'll chat reef with Joe. So until then, be safe out there, and we will see you next time.